Enzymes. So what are enzymes? First of all enzymes are proteins that function as biological catalysts. There are some enzymes which are not proteins, and those are the more recent ribozymes, which are RNA-based enzymes. The enzymes are essential for reactions to occur in the living as well as in the dying cell. They have a profound effect on the quality and the deterioration of food. And they have some very positive reactions which are used and exploited in food processing and beverage processing, other than in the textile and pharmaceutical industry. One of the major properties of enzymes is they're highly specific. So if an enzyme acts on A, it cannot act on B, and so on. The reactions are very rapid. Some of the reactions can be complete in nanoseconds. Very small amounts of the enzyme are required as catalysts, and you can control these enzymes by controlling the pH, the temperature, the substrate concentration, and the enzyme concentration. Since most of the enzymes are proteins and they are isolated from natural sources, they are non-toxic. Several enzymes have been given GRAS status. What is GRAS? G-R-A-S, that is granted as safe. So enzymes are specific for the reaction they will catalyze. So you see a specific substrate will act and a specific enzyme will act on this substrate. All the enzymes are reusable like other catalysts, and most of the enzymes, if not all, end with the script ASE. That is, they're either sucrase, which acts on sucrose, lactase, which acts on lactose, maltase, which acts on maltose, and oxidase brings about oxidation. So simple enzymes are whole proteins other than the ribosomes. You also have complex enzymes, which are a protein plus a relatively small organic molecule which is required for its activity. So a holoenzyme or a complete enzyme is the protein which is called as an apoenzyme and a cofactor, which could be the prosthetic group or it could be the coenzyme. And in Friday's class on vitamins, especially the vitamin B complex, we have discussed how most of the B complex vitamins serve as coenzymes in several oxidation reduction reactions, carboxylation reactions, etc. The prosthetic group is a small organic molecule. It could be a coenzyme or it could be a cofactor, such as a metal. Several enzymes require metal as the cofactor, or metal is bound to the protein and it serves as an enzyme. So in this next table, I've given you some examples of cofactors, of the coenzymes required, and some of the cofactors required. So thiamine pyrophosphate, TPP, is required in the oxidative decarboxylation of pyruvate dehydrogenase. FAD or FMN is required as for monoamine oxidase. NAD, the cofactor of niacin, is required for the lactate dehydrogenase. Pyridoxal phosphate it is the cofactor in glycogen phosphorylase, and you have tetrahydrofolate, which is involved in one carbon transfer and acts as a coenzyme for thymidylate synthase. Several metals serve as cofactors. Important to remember are zinc in carbonic anhydrase, zinc again in carboxypeptidase, magnesium in hexokinase, nickel in urease, okay, and you have manganese in super dismutase. So several enzymes are serve as cofactors, or they serve as prosthetic groups in the enzymes. So, what are the characteristics of enzymes? They characterize a reaction, and the reaction rate goes several million times faster, 10 to the power of 16 times faster than an uncatalyzed reaction. The second advantage is they can be used in mild conditions. So these can be used at low pH, at low temperature. They have a reaction specificity so that only the desired product is produced. And the enzymes in the human body can be regulated. The factors that influence enzyme activity are the pH of the medium, the temperature of the medium, the concentration of the enzyme, and the substrate concentration. So, how do enzymes work? The way enzymes work are they weaken the bonds by lowering the activation energy. So if you look at this diagram on the left-hand side, this is an uncatalyzed reaction where you see that if this is the reactants and the products have to be produced, the reactants have to be to react and they have to be taken to a higher energy level, what we call as the energy of activation. And then the products are formed. If the same reaction is catalyzed in the presence of the enzyme, you see that this energy barrier, that is the activation energy barrier, will be reduced several fold and the products will be formed. So basically, enzymes act by lowering the energy of activation, that is lowering EA. So this is a better picture to show you how the lowering of the energy of activation takes place. So if a ball has to be pushed to across a mountain, and if this is the energy used in the absence of the enzyme, in the presence of the enzyme, you see there is not much energy required. Here, this reaction shows you the action of lactase on the enzyme lactose or beta galactose, which breaks down lactose into glucose and galactose. So basically, what you need to understand is that enzymes lower the reaction's energy of activation. So enzymes, there are several types of enzymes, and enzymes have been classified into 6 different groups. The 6 different groups to which they have been classified by giving the numbers. These numbers are also very specific for the enzymes. Group 1 is the oxidoreductases and encompass all the dehydrogenases, all the oxidases. An example of this is alcohol dehydrogenase, which actually converts ethanol into acetaldehyde. Group 2 is the transferases, and these transferases are involved in the transfer of a group. So, hexokinase, you see, is the transfer of a phosphate group from ATP to glucose. So, the transferases, this is a transferase, and all transferases will begin with the number 2. The third group is the hydrolases. The hydrolases use water in their reaction and they break down a bond to give you the products. So the example here is of carboxypeptidase, which breaks the carboxy terminal amino acid of a protein to give you the peptide, which is shortened by one amino acid and the C terminal residue. Number 4 is the lyases or the synthases, which are involved in the formation of bonds or thing. So pyruvate decarboxylase, pyruvate is converted into acetaldehyde. And it has the EC number, it starts with 4. Then you have the isomerases, which are involved in the conversion of cis to trans isomers or into the epimerases, what you have seen in the carbohydrates, or even glucose isomerase, which converts glucose into fructose. And the last group is the ligases, which will actually make the bonds. So you have pyruvate carboxylase, where pyruvate is converted into oxaloacetate, and this is a carboxylation reaction. So the ligases make the bond isomer, and excuse me, I said liases make the thing, but they actually break the bonds. And here the carboxyl group, it's a decarboxylation, it breaks the bond. So liases bring about, they break the bonds. Now, if you look at the enzyme nomenclature, they are all appended by A's. So you have alcohol dehydrogenase, urease for hydrolysis of urea, DNA polymerase, which brings about the polymerization of nucleotides. Then you have several enzymes which do not end with ASE but have enzymes such as pepsin, trypsin, chymotrypsin, lysozyme ends with the zyme. So these enzymes were named so because they actually catalyzed, their reactions were known much before the other enzyme. But you cannot keep naming these enzymes as and how you please. You can have the names as common names or trivial names. So therefore, the International Union of Biochemistry and Molecular Biology, this is an apex body for biochemistry. They have made rules which are based on the 6 major classes of reactions catalyzed, where the numbering of the enzyme is based on the rule, and you have a major class, then you have a subclass, and you have subclasses, just like how you do so in taxonomy. So in the case of the enzymes, the enzyme initially has got a systematic name like ATP D-glucose 6-phosphotransferase. The trivial name is hexokinase. This is an enzyme which adds the phosphate group to a sugar. So it could be glucokinase, fructokinase, for any hexose. So it adds the phosphate group using ATP and magnesium to form the phosphorylated sugar. So it has the EC number 2, E full stop, C full stop, 2.7.1.2. Each of these is significant. So EC stands for the Enzyme Commission. Two, so EC stands for the Enzyme Commission. The first number stands for the class. This is transferase, and if you recollect, we saw that transferase belong to class 2. 7 is the subclass, and 1 is the subclass, and 2 is the individual entry. So therefore, hexokinase has the enzyme EC 2.7.1.2. So in any professional document, even though you write hexokinase, within parentheses, you must put the EC number. So if we look at the hydrolases, which use water as one of the substrate, you see the EC number for hydrolases is 3. So 3.1 is acting on ester bond, 3.1.1 is carboxyester hydrolase, and 3.1.1.3 is triacyl glycerol lipase. That means this is a lipase which breaks down the TAGs or the fats and oils in the presence of water to give you 1,2-diacylglycerol and fatty acid. So in the same way, the enzymes that you have had a chance to be associated with, the glycosylases, again, which use water and are used in the breakdown of sugars, have 3 for hydrolases, 3.2 represents glycosylases, 3.2.1 is a glycosidase, hydrolyzing It's an enzyme hydrolyzing O and S glycosyl bonds. And then you have 3.2.1.1 is alpha amylase, which can break down starch. And then you have 3.2.1.2, which is a beta amylase, 3.2.1. The enzymes with the EC number 3.2 are glycosylases, and in that you have 3.2.1, which is a glycosidase. And you see all the enzymes which belong to this group bring about the hydrolysis of carbohydrates or the polymers of sugars. So what is the enzyme reaction? So if you look at the enzyme reaction, conventionally the enzyme reaction is represented by S, which is the substrate, and that is converted into the products, which is catalyzed by the enzyme E, which does not change at all, or it remains unchanged. So enzymes are highly specific or selective, as we say they are selective, and the specificity is controlled by the structure of the enzyme. And as we will see that the enzymes react with the substrate by a unique mechanism known as the induced fit theory, or as a lock and key mechanism. How a single key fits a lock, similarly a single substrate for the enzyme. The enzyme is very, very specific for the substrate. So enzymes are substrate specific. So you see here, in the case of the breakdown of sucrose, the enzyme is sucrase. It will break down glucose into fructose. However, sucrose cannot break down maltose into glucose and glucose. So also sucrose cannot break down lactose into glucose and galactose. That is because the enzyme sucrase has got a substrate binding site in which only sucrose, the disaccharide, can fit, and we have the groups in the protein which will act upon the sucrose. So basically, if you look at an enzyme substrate reaction, so you have a substrate, it reacts with the enzyme, there is an induced fit change in the structure. This glycosidic bond is broken in the presence of water. So you have the two products, glucose and fructose. The products, glucose and fructose, are released, and the enzyme now again is available to take on the second substrate molecule to break it down into glucose and fructose. So this reaction cycle continues in the enzyme catalyzed reaction. So the enzyme is continuously being recycled, or there what we call as the turnover. So it is being turned over. Factors that control the action of enzymes, we will see how the substrate concentration controls the activity of an enzyme, the enzyme concentration, the temperature, the water content, the pH, inhibitors, and if there are any alteration in the substrates and or the products. So if you look at the effect of substrate concentration and you look at the graph, if you study the effect of the substrate concentration, which is on the x-axis, and you measure the velocity of the reaction on the y-axis, initially you will see that the velocity is directly proportional to the substrate concentration, or it follows a first order kinetics. So initially, as you increase the substrate concentration, you will see that the velocity increases and the product increases. However, as the substrate concentration reaches a maximum, you will see that the velocity has plateaued off, and however much you increase the substrate concentration, the velocity of the reaction will remain the same. And here what we say is the enzyme follows a zero order kinetics. So initially at low substrate concentration, it follows first order kinetics, and at high substrate concentration, you have zero order kinetics. That is, however high you feed the substrate, you will find the rate of the reaction will remain the same. And each of these reaction rates is catalyzed by a constant. So if you have an enzyme which reacts with a substrate to give you an enzyme substrate complex, the forward reaction is catalyzed by a rate constant K1, and in the enzyme substrate complex is broken down into the enzyme and product by a rate constant K2, and the reverse reaction is by K minus 1. So this reaction curve is very specific to enzymes, and this is known as the Michaelis-Menten curve, because it was Michaelis and Menten who actually found, or who were able to show, and who were able to explain mathematically this equation, that initially it is first order and finally it is zero order. What we need to understand from this graph are three terms: Vmax over here, which is the maximum velocity that the enzyme can produce under the conditions tested; Km, which tells us the affinity of the enzyme for its substrate. It is the substrate concentration at half the maximal velocity. So if you have a very low Km in the order of 0.001 millimolar compared to 0.01 molar, so the enzyme is more specific. So the affinity for a substrate is much higher when the Km is lower. And the catalytic efficiency of the enzyme is described by the term Vmax by Km. So this is Michaelis-Menten equation to explain the velocity of a reaction. So the effect of substrate concentration, just to show you it in a better thing. So here you have first order and here you have zero order. So at low substrate concentration, enzyme and substrate determine the rate, and at high substrate concentration, only the enzyme determines the rate of the reaction because it is saturated. So what is the effect of enzyme concentration on the reaction rate? What we see is as you increase the enzyme concentration, the velocity of the reaction increases linearly. And this is what you are expected to see. So if you have a velocity of 1 at this concentration, at 2x concentration of the enzyme, the velocity should have doubled. At 3, it should have tripled. At 4, it should have, it's multiplied by 4. If the increase in the velocity is not proportional to the enzyme added, then it indicates that you have an inhibitor in the reaction system. So the effect of enzyme concentration on the reaction rate is used to check for the presence of inhibitors in the preparation. So a parameter on which the enzyme activity depends greatly is the temperature. So it is observed that if you plot the reaction velocity against the temperature, what you get is a Gaussian curve, where as the temperature increases, the velocity of the enzyme reaction increases, and then after a particular temperature, you will see that the activity decreases, or that is because thermal inactivation takes place. So this particular temperature is known as the optimum temperature, and you determine the optimum temperature and use it. Different enzymes have different optimum temperatures. Some work at 15 degrees, some work at 20, some work at 37. The optimum temperature for all enzymes in the human body is 37 degrees Celsius. So it is very important to determine the optimum temperature of an enzyme before using it in food processing, so that you do not have any undesirable effects, or it does not get inactivated. Because we have seen that proteins can be inactivated at very high temperatures. The next parameter, which is very important for the enzyme, is the pH. So the effect of pH is drastic on the activity of the enzyme, because the enzyme is made up of different amino acids, and the mechanism of action depends on what is the state of the amino acid at the active site, whether it is protonated, whether it is deprotonated, or whether it has, whether the guanido group has got NH plus. Most of the enzymes have an optimum pH between 4.5 and 8. So if you plot the activity of the enzyme versus the pH, or the reaction rate with the pH, what you will see is the activity increases, reaches a maximum, and then decreases. And generally it decreases either by inactivation or some change in the side chains of the critical amino acids at the active site. For this reason, pH control of foods with undesirable enzymes is important. So every enzyme is characterized by a Vmax, it is characterized by a Km, it is characterized by an optimum pH, and it is characterized by an optimum temperature. So if you heat the enzymes beyond the optimum temperature, because they are proteins, they get denatured. You have seen what happens to proteins. They get denatured, they lose their tertiary structure, and they sometimes they lose their secondary structure also, and they are inactivated. Just like you have seen that the egg white, if heated, which is transparent, will become opaque. The denaturation will change the shape of the enzyme. It Enzymes can be denatured even by change in pH. You know that pH also can cause denaturation of proteins. So the in food processing and in the food, the water activity, just this morning we discussed about water activity. So the water activity can influence the activity of an enzyme in several ways. It is very critical, especially for conversion of substrate to product. So if you look at the lipase reaction, if moisture is present in the food, then lipase can act on the fats and break down them into fatty acids and glycerol. If the water activity is critical to solubilize the substrate, and it is critical for the flexibility of the enzyme structure, because the enzyme is a protein. And it is, if you recollect the last figure that I gave you on water activity varies, can be varied in fluid to slow down the enzyme activity. So if you decrease the water activity, decrease the water activity by adding sugar or by adding salt, you will find that enzyme activity will be decreased. So here is enzyme 2 and enzyme 1. You see they have their activity depends on the AW. The enzyme also, activity of an enzyme will depend on what are the other factors which are present in the solution, and those are inhibitors. So it is the effect of enzyme concentration clearly shows you that if there is an absence of inhibitors, the enzyme, the velocity of the reaction is directly proportional to the concentration of the enzyme. However, sometimes we may not require the enzyme to act for the activity to be optimum, and you can inhibit the enzyme by adding what are called as inhibitors. For example, if you observe when you cut an apple or you cut a potato, it becomes brown immediately. That is because the enzyme polyphenol oxidase has caused the browning. Now, to prevent the browning, you can remove one of the substrates. For example, oxygen is the substrate. So you can put it into water to remove the oxygen, or you can add sodium chloride, which is an inhibitor of polyphenol oxidase, or you can add citric acid, which is an inhibitor of polyphenol oxidase. So therefore, different inhibitors can be added. In terms of the inhibitors, we have three types of inhibitors which are used. One is the competitive inhibitors. The name itself suggests that it will compete with the substrate. So the structure of the inhibitor is very similar to the substrate. And so the enzyme can bind either to the substrate or to the inhibitor. It cannot bind to both the substrate and the inhibitor at the same time, because both will compete for the same place in the enzyme structure. The second type of inhibitor is a non-competitive inhibitor. It actually binds to a site other than the substrate binding site, but it will cause a change, a conformational change in the enzyme structure, preventing the substrate from binding. So you have non-competitive inhibitors which bind to another site other than the substrate site. They change the conformation of the enzyme and prevent the actual substrate from binding. And then you have the uncompetitive inhibitors which bind to the enzyme substrate complex. They cause a conformational change, and they prevent the enzyme from these inhibitors, these three inhibitors, competitive inhibitors, non-competitive, and uncompetitive inhibitors, are reversible. That is, if you add more substrate, you can reverse this inhibition. There are irreversible inhibitors which completely change the structure of the enzyme or denature the enzyme, and even if you remove the inhibitor, you will not be able to use the enzyme again. A very good example of this is that of mustard gas or nerve gas, which was used in the world wars. So mustard gas or nerve gas are irreversible inhibitors of acetylcholine esterase. So when nerve gas bombs were thrown, it inhibited the acetylcholine esterase, and you know how important acetylcholine esterase is for nerve transmission. So because of the inhibition of the acetylcholine esterase, it caused paralysis in most of the warriors. A similar compound, similar to nerve gas, which is a pesticide which is called malathion, is used to inhibit the feeding predators, that is the insects in the crop plants. So here also the target is the acetylcholine esterase enzyme of the predators, and it paralyzes the insect predators, and they die. So irreversible inhibitors will completely kill the enzyme, and they cannot be brought back into the original condition, whereas reversible inhibitors can be revived by changing the conditions. Some food use for these are in terms of flavor, odor, color, and toxicity problems. So this is the example to show you. In the case of a competitive inhibitor, the competitive inhibitor will mimic the substrate. So therefore, this is the substrate binding site. The inhibitor will bind to the same site, And what it does is it will not change the Vmax, but it will change the Km. An uncompetitive inhibitor is an inhibitor which binds to the enzyme substrate complex and causes a change. So it causes a change in the Vmax of the reaction as well as the Km. I'm not going into the detail of the equations for these because they are a class by themselves to derive and show you how Vmax changes or how the Km changes. These are two parameters by which an enzyme is characterized. So in a non-competitive inhibitor, the enzyme inhibitor binds at a different thing site, causes a conformational change, and does not allow the substrate to bind. So here what happens is the Vmax will change, but the Km will remain the same. So just like how you have enzyme inhibitors, we have enzyme activators. Activators which are required to help the enzymes work faster or to increase the reaction rate. Inorganic compounds and vitamins are activators. So you have an activator, it has a binding site, it makes the active site more conducible for the substrate to bind. So enzyme activators also act like the inhibitors by allowing the enzyme reaction to go much more quickly. So these enzyme units, as I said, enzyme depends on the substrate concentration and we have a velocity. So how is this enzyme activity represented? So one international unit of enzyme activity is defined as the amount of product formed per minute at constant temperature and pressure. So it will be expressed in terms of micromoles of substrate converted per minute or micromoles of product formed per minute. The international unit actually is the katal, which is represented by KAT. It is defined as the amount of enzyme which converts 1 mole of substrate per second. So here it is in terms of moles per second. And 1 katal is equal to 6 into 10 to the power of 7 micromoles per minute. But in any enzyme activity, the temperature and the pH must be defined. Without defining the temperature and the pH for this activity, the enzyme unit is meaningless. So what is the mechanism by which enzymes act? There are two hypotheses put forward. One is that called the lock and key mechanism. That is, it is said that in every enzyme you have, it is like a key, it fits into one lock. Although this may not be true because many keys can be put into a lock, but generally there is a single key which fits into a lock. So this is the enzymes react by a mechanism known as the lock and key model. So you see they fit in exactly into the active site of the enzyme. And then you have another hypothesis, which is called the induced fit model, where it says that you have an enzyme, and when the substrate comes and binds, the enzyme undergoes a conformational change when the substrate binds so that it can actually accommodate the substrate. That is an induced fit model. It changes its shape and then you get the enzyme product complex and the product is released and the enzyme is ready for the second cycle. So two hypotheses are there, the lock and key mechanism and the induced fit model. Now let's come to examples of the enzymes. What I discussed previously were generally the properties of the enzymes. Now let's look at some of the examples of enzymes which are used in the food industry. In milk, we have lactase or beta galactosidase. which is used to break down the lactose into glucose and galactose. Alkaline phosphatase in milk is very useful to tell us how well the milk is pasteurized. So if you get alkaline phosphatase activity, that means it has not been, if you are able to measure the alkaline phosphatase activity, that means it has not been pasteurized. Lipases in milk can break down the fats. It is used to produce flavors in certain cheeses. In fresh versus canned pineapple, there is an enzyme in pineapple called bromelain, which breaks down gelatin into jello. Meat sometimes is very tough, cannot be cooked if it is an older animal, but bromelain is a protease from pineapple, phycin is a protease from figs, and papain is a protease from papaya, which can be used to break down the peptide bonds in the meat and tenderize the meat. When you do the blanching of vegetables, when you put it into boiling water and just remove it, it's called blanching. So what happens in blanching is actually you are inactivating the enzyme catalase and peroxidase, which can cause discoloration in the enzyme. We will see how you can get clear and cloudy apple juice by using pectinase enzyme, and you can also see the pressing from mandarin oranges. When you cut onions, you see that your eyes burn. Why is it your eyes burn? That is because we have the allinase as a thing enzyme, which is in one compartment, and the substrate, which are sulfur containing enzymes, sulfur containing substrates in another compartment. When you cut the onion, these two come in contact, and you have these very pungent sulfur compounds which are released, which are lacrimatory. That means they produce tears. So allinase causes the tears. So hydrolases, which are important in the food industry, most of the hydrolases belong to the enzymes that will break down starch. So alpha amylase, we have seen, is the enzyme will break down the alpha 1,4 glycosidic linkages within starch. Beta amylase hydrolyzes the glycosidic bonds from the non-reducing end of starch. Glucoamylase hydrolyzes alpha 1,4 and alpha 1,6 glycosidic bonds in starch, and pullulinase hydrolyzes the alpha 1,6 glycosidic bonds in pullulan. So in the baking industry, these glycosidases are very important. Alpha amylase is added to the baking floor to dextrinize, that means to break down the starch granules into smaller molecules called dextrins, which are then hydrolyzed to maltose. And then when you have added yeast, the yeast can ferment the maltose to give you carbon dioxide, which is essential for the rising of bread or the spaces that you see. So in baking, the amylases are very important to gelatinize the starch, and they play a very important role in the final texture of the quality of bread. And the amylases added to bakery products can also minimize staling. So another example of an enzyme that is used in the baking industry is an enzyme called glucose oxidase. You know, potassium bromate has been banned and it cannot be used in food. Earlier, it was used in wheat flour to strengthen the dough mixture and to enhance the elasticity. But since it is banned now, you have to find an alternative, and the alternative which is found is that of okay, the alternative that you have found is glucose oxidase, and it forms gluconolactone and gluconic acid, and glucose oxidase can actually bring about the same properties as that of potassium bromate. So enzymes important. The alpha amylases are very important in the brewing industry because alcohol is produced from maltose and malt. So the high level of amylases in the barley malt result in the production of carbon dioxide and ethanol, and they are also used to convert starch into maltose. So the amylases are very important in the brewing industry also. Not only are they important in the baking industry, but they are important for the brewing industry also. So the enzymes are useful for making the high corn fructose syrup, because if you see, high corn fructose syrup is made from corn starch. So initially in the preparation of high corn fructose syrup, gelatinization of the starch is brought about, and then bacterial alpha amylase is added at pH 6.5 and at very high temperature. So it breaks down the starch into dextrins. Then a second group of enzymes called the glucoamylases is added to produce glucose syrup. And then you add a third enzyme, the glucose isomerase, to convert it into fructose. And what you get is high fructose syrup. If you add pullulinase to the and beta amylase to the dextrins, what you get is a high maltose syrup. So depending on which end product you require, you use a different pH and temperature regime. So that is why it is important. And as I said, every enzyme has got an optimum pH and an optimum temperature at which it can convert the substrate into a product. So the glucose isomerase is an enzyme isomerase, so it converts glucose into fructose. So in the last step of this, the glucose syrup to the glucose syrup, the glucose isomerase is added, and you get the fructose syrup. High fructose syrup is very economical, and it is used in a large number of food industries. So the reason being that fructose has a relative sweetness of 150 to 70 compared to sucrose, whereas the high fructose syrups have a relative sweetness which is almost the same as that of sucrose. So high fructose corn syrup of 42% is 90% as sweet as sucrose, and high fructose corn syrup with 55% is the same as that of sucrose. And you see, high fructose corn syrup with 90% is of fructose is 120 to 160 times the. So the number of products in which high fructose corn syrup is used ranges from baby foods to canned fruits to frozen fruits to ice creams, jams, jellies, confections, etc. So therefore, the enzyme has a very important role in the high fructose corn syrup industry. So invertase is the enzyme which converts the sucrose into glucose, free glucose, and fructose. And also it is very popular in the confectionery industry because invert sugar, that means sugar, sucrose, which is converted into glucose and fructose, equal quantities of glucose and fructose, is much sweeter than sucrose, and it is popular in soft candy fillings. Beta galactosidase, or this lactase, hydrolyzes the beta 1,4 linkage between galactose and glucose. It increases the sweetness because lactose is not as sweet. When it is broken down, you get glucose and galactose, which are sweeter. It is used in the dairy industry to prevent the crystallization of ice cream, because sometimes when ice cream is kept for a very long time, what you get is crystals, which is not very nice on the tongue. So, beta galactose is also used to produce lactose free products, especially for those who are lactose intolerant, that is, those who cannot digest lactose because of the lack of the enzyme beta galactosidase in the intestine, and they will not be able to drink milk. So, you have lactose free milk, lactose free yogurt, and a large number of lactose products. Pectinase is another enzyme which finds wide use in the fruit and vegetable industry, especially for the degradation of pectic substances. So this is freshly squeezed orange juice, and on keeping the orange juice, what you get is the loss of the cloud in the citrus juice, and this is a big problem. That is because the enzyme pectin methyl esterase is present in the orange juice, and it can break down the pectin into the pectic acids and to the methyl esters. So you have to prevent this loss of cloud in the citrus juice, and what you do is you have to inhibit the pectin methyl esterase by heating it to 90 degrees for 1 minute, and then you will see that your orange juice remains with the cloud. On the other hand, here in the case of orange juice, pectin methyl esterase has to be inhibited. However, in the case of apple juice, to be able to squeeze more juice out of the apple, the pectin methyl esterase is used. So the pectinase is used to break down the apple cell walls and to enable greater juice extraction. And also, apple juice, you would like to drink a clear apple juice and not a cloudy apple juice. So it is used to break down the pectic polysaccharides which are present in the apple juice. Proteases, as I seen in the earlier slides, they are used to break down the peptide bonds. So papain, which is got from the raw papaya, if you look if you have plucked a papaya, you will see the milk which oozes out from its stem. That is a very rich source of papain, and it is sold. It has an optimum pH between 3 and level. You can use this at very low pH. It is used as a meat tenderizer, especially to soften the muscle tissue of the animals. Then you have to use it in very low amounts, and if you mix raw papaya with jello, it will not form a gel. Other tenderizing enzymes are phycin from figs and bromelain from pineapple, including some other microbial proteases. But however, since phycin, bromelain, and papaya are from natural sources, they are preferred over the microbial proteases. Um, and so the papain is also used to break down the tannin peptide complexes in certain juices, because it can break the bond between the tannin and the peptide. So other important enzymes which are also digestive enzymes are trypsin and chymotrypsin, which are found in animal pancreas produced. They are also used to tenderize the muscle foods, and they are used to make protein hydrolysates for the food and beverage industry, and they are active between pH 7.7 to 9. Pepsin, in addition to being used as a tenderizer, is used in cheese making, in chill proofing of beer, and also in making protein hydrolysates. You see, in the beer industry also, if you see beer on cooling will become cloudy, just like the juices, and nobody would like to drink that type of beer. So papain is added to break down the proteins and the protein complexes in them. Chymosin is a very important enzyme which is used in the cheese industry also. It is also known as renin, and it is found in the fourth stomach of the young calf. It was very expensive and it was inhumane to kill the calves to get the chymosin. So now a recombinant chymosin having the same sequence and forming the same protein like chymosin is expressed in aspergillus and used. It has a very specific activity that it actually only breaks a single bond in kappa casein, one of the many proteins that make up milk. Renin also is the first enzyme in our stomach which will cause the clotting or the breakdown of the milk protein. But in the industry, the chymosin or the renin is used to make cheese, and also it is used for casein, to prepare casein hydrolysates. So microbial proteases, there are several microbial proteases which are used in the food industry. Most of them are fungal proteases from aspergillus, and a protease cocktail from aspergillus is used to break down the bread proteins to reduce mixing time. Some proteases are added to produce flavor and texture, and some fungal proteases are used to tenderize meat. Among the bacterial proteases, subtlysin is from Bacillus subtilis is popular, and it is used in combination for papain in beer chill proofing. Lipases, again, are the ones which hydrolyze the ester bond of triethyl glycerols. Some lipases are used to produce different flavors like the butter flavor and different flavors required in the food industry, which depend on the fatty acid composition. The lipases are very specific for hydrolyzing either the SN1 and SN3 or the SN2 bonds. So some of the lipases known in the food industry, some of them are bad because we have seen when looking at the lipids, if it hydrolyzes and free fatty acids are produced, these free fatty acids cause rancidity as well as they increase the peroxide value. And some lipases are good when they are used in fermented products, extremely important in the ripening of cheese and dry sauces, and the short chain fatty acids released from milk are produce characteristic odor and flavor of these products. Uh, the lipids, the lipases are used to modify the properties of lipids, especially in the fat industry. So they are used to produce mono and diglycerides, and they are also used to produce DAGs. And we have seen how the lipids can form a phospholipid bilayer, and they are able to take in the fat into the hydrophobic part and hold the water in the hydrophilic part. Among the oxidoreductases, the lipoxygenases which are present, especially in soybean, are important. They are produced in a large variety of plants, and they bring about the oxidation of fatty acids to give you linoleic acid, linolenic acid, and arachidonic acid. So the desirable effect of this is the lipoxygenases is used to bleach wheat and soybean flours. It also contributes to SS bonds. But undesirable is that the lipoxygenases bring about lipid oxidation. They break down lipids and they produce hydroperoxides, which give an off flavor. They also can oxidize the chlorophylls and they can also oxidize the vitamins. However, they are used in the bleaching of wheat and soybean flours. A very important enzyme in the food industry is the polyphenol oxidase. This polyphenol oxidase again has the goodness as well as the badness. It is found almost universally or ubiquitously. It is found in many plants, animals, insects, microbes, etc. It catalyzes the oxidation of a polyphenol, a monohydroxyphenol into a dihydroxyphenol, and the dihydroxyphenol or the catechol is broken down into quinones. These quinones polymerize and they form melanins, and these cause undesirable browning, such as the browning of apple juice, the browning of apple juice, browning of apple slices, browning of bananas, mushroom, shrimp, lobster, and also the freckles that you see on your face. Up to 50% of economic loss is observed. But we have desirable browning, where the browning of raisins, the browning of cocoa, prunes, as well as dates, tea, and coffee is required. So here you have the good, and here you have the good, and here you have the bad. So in the case of undesirable browning, it can be inhibited. So how do you inhibit the enzyme? You can remove one of the substrates, that's oxygen. So if you cut a potato, or if you cut an apple, or if you cut a brinjal and you put it or steep it in water, it will not brown. That is because you have removed oxygen. You can also add lemon juice, bring down the pH to less than 4.5. You will see that browning is prevented. You can also add ascorbic acid to prevent the browning. You can add bisulfites to prevent browning, and you can also add EDTA to prevent browning. But polyphenol oxidase, the control of polyphenol oxidase is very important in the fruit and vegetable and beverage industry. So here is the browning that you see in the shrimp. So this is because of the polyphenol oxidase. So here is a browning of the apple. Here you have browning of the potato slices, and so you have to inhibit the enzyme. Although now by genetic engineering also we can prevent the browning by expressing the enzyme by a technology called antisense mRNA technique. So the enzymes are also important to the chocolate industry. So invertase is used to create soft-centered chocolates. The center initially contains sucrose, that is cane sugar, which is very hard. And so if you add invertase to the sucrose, it converts it into glucose and fructose, making the center soft and runny. So the applications of the enzymes in the food industry are numerous. There are numerous enzymes. I may not have covered all the enzymes. I've tried to cover as many as possible. And enzymes are also used, the invertase enzyme is also used to make the drinks sweeter, and you will find that it is used in slimming foods, because if you have high fructose syrup, you can add less because it is sweeter. And in sports drinks, it is used because it has the fructose which is there, takes a longer time to release the energy. And so in the endurance sports drinks, you will find the invertase converted sucrose into glucose and fructose. So food enzymes, also some of the enzymes cause denaturation. Uh, Enzymes in food are denatured by heat. For example, if you take about the blanching of vegetables, vegetables and other like capsicum also, this actually inactivates the catalase and the peroxidase, which are responsible for spoilage of the food. And so even if you're making the frozen beans, frozen vegetables, you do an initial step of blanching before freezing. So the denaturating you can do by changing the pH. You know that if you make the apple juice a little more acidic, you can prevent the browning of the apple juice by adding slight lemon juice. You add citric acid to shrimp, you can prevent the browning of shrimp. And salt is also used to inactivate some enzymes. For example, in the canning of foods also, salt is added. It brings down the water activity, and when it brings down the water activity, the activity of the enzyme is inhibited, and sometimes the salt causes osmotic shock to the bacterial cells, and the bacterial cells will not grow. So therefore salt is used to prevent spoilage in canning, and it is also used to prevent certain enzymes, so that there is no spoilage. So the enzymes used in the meat and the seafood products, we have already looked at papain, phycin, and bromelain, which are used for tenderness. This improves chewability. It reduces the bitterness. These enzymes like papain, phycin, bromelain, and other proteases are used to produce the hydrolysates of the offals or unused parts of the meat. They are also used to enhance the flavor in milk. The enzyme transglutaminase is used to restructure the protein. So it is used in making surimi type of products with the trimmings where the proteins are cross-linked. They are also used to produce the umami taste. That is, the glutamic acid is there, which gives it the umami taste or the meaty flavor. And here the umami taste is represented by the glutamate and 5-ribonucleotides GMP, that is guanosine monophosphate and inosine monophosphate. So enzymes have got a large number of applications, both good and bad, in the food industry. And last, we'll see that not only is it used in the food industry, but it is used in diagnostics, in the pharmaceutical industry. And it is known that people who have got pancreatic disease, they are given lipase. If you have a stroke and you have a blood clot, the enzyme streptokinase is given so that it dissolves the clot. Liver enzymes, if they are found in very high quantities, SGOT and SGPT, that indicates liver damage. Urine from diabetic contains glucose. An enzyme in Clinistrips, which is mobilized on the strip called glucose oxidase, breaks down the glucose, and it is used to detect the sugar. So enzymes have wide and varied applications, both in the food industry, in the medical industry, in the pharmaceutical industry, etc. So the next class will be on plant pigments.